Hello everyone. So this is a continuation of reproductive system, that is genetics. Okay, what do you mean by genes, DNA, etc. So this is how the genes and the genetics will work out. Okay, so how our offsprings will look. So the offsprings, that is the next generation, will have the combined characters of both from father and mother and from the ancestors. So how it is carried means with the help of the genes. So basic concept of genetic. So mainly Mendel, that is Gregor John Mendel. So he is the called as father of genetics. Okay. So Mendel's observation led to two laws regarding the transmission of hereditary characters from generation to generation. So first law is the principle of segregation. So that is two alleles segregate randomly from each other during the formation of gametes. That is the two things will separate each other. So it means that each gene will have two copies, that is called as alleles, and each parent will have one copy to the child. Okay, so parents, that is capital A and small a and a, so it will be divided into two alleles, A and small a. So which will give either 2a or capital A small a, or either 2 small a. So this is how it is carried out. Okay, so next is the second law. So principle of independent assortment. So two genes will assort independently of one another in gamete production. Okay, so alleles means are different version of the same gene. Homozygous, it will refers to individual with two identical alleles. Heterozygous means two different alleles. Genotype means specific allele composition of an individual, and phenotype means outward appearance of the individual. Okay, so genetic material or genome. So a genome can be defined as a genetic material of an organism. It consists of DNA or either RNA in case of viruses. The genome includes both genes and coding gene and non-coding DNA, and the genome of the mitochondria and chloroplasts. So cytogenetics, it is the study of chromosomes. So cytogenetics, it is the study of chromosomes and their role in their heredity. So cytogenetics focuses on studying the structure, composition of chromosomes as well as diagnosis chromosomal abnormalities associated with diseases. A karyotype will refers to the number and appearances of chromosomes in each nucleus of the eukaryotic cell. It is also refers to complete set of chromosomes in a species. A normal human karyotype will contain 22 pairs of autosomes and 1 pairs of sex chromosomes. Okay, so autosomes means which will determine the body characters, and sex chromosomes will determine the sex of the fetus. So chromosomes is a thread-like structure. It consists of DNA molecule packed tightly and coiled around the specific proteins called as histones. So chromosomes are located in the nucleus of the eukaryotic cells. Okay, so normally the DNA is a double helix structure. So here it is surrounded by a histone protein. So it will be converted into chromosomes. So structure of a chromosomes. So chromatids are identical parts of each chromosomes. So these chromatids are held together at a point named centromere, which can be located at any point along the length of the chromosome. Okay, so telomere, it is the region of the repetitive nucleotide sequences. So telomere is located at each end of the chromosome. It protects the end from the chromosome with the deterioration or from fusion with the neighboring chromosomes. Okay, so in this diagram you can see what is chromatid and a short arm, long arm. So P arm is the short arm of the chromosome and Q arm is the long arm, and the middle portion is called as centromere and the extreme portion is called as telomere. Okay, so next is the chromosomal abnormalities. So almost every cell in our body will contain 23 pairs of chromosomes. So total of 46 chromosomes. So 22 pairs are called as autosomes and one pair of sex chromosomes. That is, the sex chromosomes will be XX in case of female and XY in case of male. So chromosomal abnormalities can be categorized as numerical and structural abnormalities. So numerical means the number of chromosomes. Okay, that is instead of 23 they will be having either reduced or increased amount. Structural abnormalities, so there will be a part of chromosomes may be missing or extra or switched to another chromosome. Okay, so this will lead to abnormalities of chromosomes. So how chromosomal abnormalities will occur means, so mainly the formation during the formation of egg or sperm, there will be chromosomal abnormality, or in case of early developmental stage of the fetus, and certain environmental factors can also play an important role in genetic errors. Example, you can have radiation, cancer inducing, a lot of things. Next is the causes for chromosomal abnormalities. So some medicines, street drugs, that is unknown drugs, alcohol, tobacco, toxic chemicals, some viruses and bacteria, some kind of radiation, certain health conditions such as uncontrolled diabetes can lead to chromosomal abnormalities. Next is the DNA. So DNA is the hereditary material in humans and almost all other organisms. So nearly every cell in the body has a same DNA. So most DNA is located in each nucleus, where the called as nuclear DNA, but in a small amount of DNA can be found in the mitochondria also. Next is the RNA. So RNA is the polymeric molecule essential in various biological roles like coding, decoding, regulation and expression of genes. Okay, so there are three types of RNA: mRNA, that is messenger RNA; tRNA, transfer RNA; and ribosomal RNA, it is called as rRNA. So messenger RNA will take the messages from the gene for producing particular type of protein, okay, to convey the message. Transfer RNA will transfer the amino acids, okay, from place to place. Ribosomal RNA links the amino acid together to form protein. So it will link the amino acids to form proteins. Okay, so these are all the functions of RNA. So the main differences between DNA and RNA. Okay, so in case of definition, DNA, it contains the genetic instructions used in the development and functioning of all modern living organisms, whereas in case of RNA, it contains the genetic information that is transcribed from DNA to RNA. Function: so medium of long-term stable storage and transmission of genetic information. So here, transfer genetic code needed for creation of proteins from nucleus to the ribosomes. So structure: RNA will be double-stranded and DNA will be double-stranded and RNA will be single-stranded. So base pairing. So I think in previous classes you have studied this. So adenine, there will be four things will be there: adenine, guanine, cytosine and thymine. So here adenine will links to thymine and cytosine will links to guanine. Okay, so here adenine will be linked to uracil and cytosine will link to guanine. Okay, there will be a change, that is instead of thymine you will be having uracil in case of RNA. So location: DNA is found in nucleus of each cell and a small quantity in mitochondria. So RNA will be found in the nucleus, cytoplasm and ribosomes too. So stability: stable in alkaline condition, RNA is less stable in alkaline condition. So propagation: DNA is self-replicating, RNA is synthesized from DNA when it is needed. So unique features: DNA is protected in the nucleus as it is tightly packed. DNA can be damaged by exposure to UV rays. In case of RNA, strands are continuously made, broken down and reused. RNA is more resistant to damage by UV rays. Okay, so this is the topics today. Remaining continuation we can see in the next class. Take care. Bye bye.